Coming up on Dr. Kiki Science Hour, we're talking about making evolution-proof drugs with Dr. Andrew Reed from Penn State University. That's up next on Dr. Kiki Science Hour.
Netcasts you love.
From people you trust.
This is Twit.
Bandwidth for Dr. Kiki's Science Hour is provided by Cachefly at CACHEFLY.com.
This is Dr. Kiki Science Hour, episode 36, with Dr. Kiki Sanford and Dr. Andrew Reed, battling the Red Queen. Hi, everybody. This is Dr. Kiki Science Hour on twit.tv, and today we are battling the Red Queen. That's right. Alice in Wonderland is opening around the country, and I can't help but compare the topic for today's show with one of my favorite characters in the sequel to Alice in Wonderland, Through the Looking Glass, the Red Queen, who had to run very fast just to stand in place. And so in line with that, what we're talking about today is the evolutionary arms race that occurs between all species on this planet, but of particular concern to humans and human health, the arms race that is occurring between infectious diseases, bacteria, viruses, those buggy little buggers who tend to make us sick and die. It's not good. And the drugs that we create to combat them and to make ourselves healthy. And today, to talk about it with us, is Dr. Andrew Reed from Penn State University, originally from New Zealand by way of the UK. And he's going to be talking with us about this question that he seems to be asking recently. I've seen him around the media asking this question of whether or not we can create evolution-proof drugs. And so today we're hopefully going to get into that a little bit. And one example of what's happening here, and I always forget a syllable in this word, the artemisinin, thank you. They are some of the most effective drugs that we have against malaria currently. And in the last year, scientists have learned that the malaria parasite is actually becoming resistant to these drugs. And so as a result, we are looking at this question of what are we going to do now? How are we going to make the drugs last longer so that scientists will have a chance to actually create the next batch of drugs that might actually be able to combat malaria? Or are there other solutions, and are there other ways around it? Can we stop this mad dash to create the next drug that's only going to work as long as the disease is not yet resistant? And can we use evolutionary principles to get around evolution? So without any more of my blathering on, I would like to welcome Andrew Reed. His lab, the Reed Group, works on ecology and evolutionary genetics of infectious disease. And he's asked the particular question, can we make evolution-proof drugs, vaccines, and insecticides? Welcome.
Thank you very much. Thanks for having me.
You're welcome. I'm really glad to be able to speak with you today, as I think this is a really fascinating subject. Most scientists tend to look on the side of, here's the bug that we're trying to defeat, that we're trying to keep ourselves from being infected by. So let's create a vaccine, or let's create something that we take as humans that will keep us healthy. But you're looking at it from a different angle. Can you explain the perception and the perspective that you're taking?
Well, we're certainly interested in vaccines and drugs that work, and our ambition is to keep them working. We're also interested in the insecticides that are used against the insects that transmit diseases like like malaria and like dengue. So what we're interested in doing is trying to figure out ways of designing or using these drugs or insecticides or vaccines in such a way that the target organism, the bug or the insect, doesn't develop resistance to our compounds quite as fast as they do now. In an ideal world, we'd like to stop their evolution, but of course that's, for the most part, very tricky. But what we'd like to do is slow down their evolution so that our drugs and vaccines and insecticides work for longer before they have to be thrown away and replaced by new compounds.
How did you get interested in this in the first place? Like, where did the inspiration for evolution-proofing drugs come from?
Well, for me personally, I've always been interested in evolution. I mean, why wouldn't you be, right? It's obviously, science has answered to why we're here. So for as long as I can remember, I've been interested in evolution. Sometime during my PhD, which was actually on the evolution of birds, I became interested in the diseases of birds. And I discovered, much to my surprise, that there weren't too many evolutionary biologists actually working on the evolution of the diseases themselves. And yet these diseases, they evolve in real time. They evolve extremely quickly in the laboratory and in the field. And I sort of naively imagined people would give you money to study and solve this evolution. So towards the end of my PhD, I got really interested in the question of malaria evolution and started working on that side of things. And for a lot of the time, I was just interested in what one might think of as generally theoretical questions of not too much applied interest. But in the last few years, particularly the last 5 or so years, I got more and more interested in whether or not we can use evolutionary theory, the ideas of modern evolutionary biology, to deal with some of the evolutionary problems that face medicine today. I think this century especially is going to be one where we are going to increasingly struggle with the counter-evolution of our bugs against the things we throw at them. And it's getting increasingly hard to come up with new compounds, new solutions, and so forth. So that was really what got me into it.
I think it's always a fascinating story to find out how people came to what it is that they study. The fact that you started out looking at birds and now you're looking at the things that infect birds or people. It's not so far of a stretch because of the whole evolutionary story that contains it.
Yeah, and I think actually a lot of the theory that we work with today in my group, a lot of it's been developed by people who were natural historians, essentially working on strange insects or strange birds and trying to figure out solutions which didn't actually seem to have much to do with anything, the sex ratio of weird wasps or something. And we use a lot of that theory in our day-to-day work with the pathogens of humans.
Yeah, so let's use that as maybe a jumping off point. A lot of the theory that say the malaria parasite that you're dealing with, you're actually thinking about the ecology of the parasite and the life cycle of the parasite and how it exists in the world before getting into these questions of how do you fight it.
Yeah, I mean, to me, it's a natural way of thinking about all organisms, and diseases are just like birds or people or mammals. They evolve, and the way you think about their evolution is to ask, well, how are they living? What sort of problems have they got to face? How do they reproduce? How can they maximize reproduction? How's evolution going to work to favor the most fit, and so forth? So you tend to think about it in precisely the way that one does for any organism, and that's quite natural because they are organisms. But that means you have to understand their natural ecology, and then you need to understand how our medical interventions, like drugs and vaccines, change that ecology.
So the medical interventions change the ecology. Can you explain how that works to somebody who might not understand that necessarily?
Well, you could think about some of these things that we do to our bugs as some of the biggest changes in organismal ecology of any sort. I mean, if you think about a vaccine, for example, a vaccine against measles or polio or smallpox, those are highly effective and they completely change the number of hosts within which those organisms can live and the types of organisms. And so, for example, in the case of smallpox, we so changed the ecology of the disease that it died out. I mean, we really just took away all its available habitat and it died out.
Huh. I think that's a really interesting comment and a point that maybe could be expanded on is the fact that when you get rid of a pathogen, you are getting rid of the habitat. So you're taking away, just like animals on the surface of the planet who we've, say, logged the Amazon or other places on the planet where they no longer have a place to live because of human interventions. This is just on a more micro scale.
Numbers-wise, it might be on a much larger scale. I mean, we're talking, billions of organisms here.
I was thinking in terms of size. Little tiny organisms.
I think in terms of numbers, and these are big. I have actually, when we've been talking about trying to understand how pathogens will evolve in response to vaccination, we often do find ourselves talking about analogies to do with habitat destruction, and quite often we find ourselves talking about polar bears. And, if ice caps vanish, the habitat within which a polar bear can exist starts to shrink down. And if it's still the same habitat, nothing else has changed, it's just got smaller, then you start to think, well, the evolution isn't really going to change much. There's no change to its ecology, there's just less habitat. But if, on the other hand, the ice caps start to melt and that reveals, let's say, more tundra, and so a larger part of the polar bear's life becomes based on rocks or even savanna-like grasslands or whatever, then the polar bear could start to evolve, and it could become, for example, a brown bear. And under those circumstances, then you've got the sort of evolution of a new organism or a new lifestyle, and that's the sort of thing we're asking about. If you're changing the world that a pathogen sees with your vaccines so that it's now a different world, it's different ecology from what they had before, what will they do? How will they evolve? Will they become nicer, nastier, easier to control, more infectious, less infectious, and so on?
When you're talking about the organism and what triggers evolution, what causes changes to happen to organisms, is there a particular point in their life that is more sensitive than another?
Well, I guess, but the way I think about it is you need to focus on what your intervention is doing. Let's say a drug that's being used to treat a person, and you ask, how is that impacting on the reproductive success of your target organism? So if we take the malaria parasite, for example, we treat a person, it's treating the blood stages, the parasites in the bloodstream. It's only treating that stage, but that's impacting on its ability to get into mosquitoes, to be transmitted on to the next person, and so on. So the trick is to ask, if you have a resistant parasite, one that can survive the drugs, or if you have the original ancestors, what's going to be the difference in how they perform? How will they do things differently, and all of that? And from that thought bubble, from that thought process, then one tries to figure out what will happen next.
So let's talk about the malaria parasite for a little while. This is one that you've done significant work on. What part of the life cycle are you focusing on in the ecology?
Actually, we have various different parts of the life cycle we're working on. There are the drugs that we are working on. Their mode of action is primarily on the parasites when they're in our bloodstream. We're also doing very significant work, I hope, on mosquitoes and trying to control the mosquito part of the life cycle, and maybe we'll come back to that in a minute. And then the vaccines that people are using against malaria parasites, or at least trying to develop against malaria parasites, they strike at various different parts of the malaria parasite. And the part that we've been most interested in is the vaccines that would protect against severe disease, so that would target the parasites when they're in our bloodstream. But the way we're thinking about it in all three cases is to ask how does the relative success of different types of parasite, by which I usually mean more or less resistant parasites or more or less resistant mosquitoes, how are they going to survive in our new world? Who's going to have the advantage, and how will it work? And that's it's a very simple thought process to start with, but it can lead into quite non-intuitive outcomes. For example, our claim that it is possible to come up with an insecticide that mosquitoes would never evolve resistance against. So you can that simple thought process of thinking, asking yourself, how would a resistant and how would a sensitive parasite reproduce relative to each other? How would their evolution proceed? That simple thought process can lead you to very interesting, perhaps non-intuitive ways of dealing with these diseases.
Do you think it's possible is the insecticide the point that we should be focusing on? Is it possible to create an insecticide that mosquitoes won't get resistance to?
Well, let me start with the idea of the insecticide in the first instance. There's a very strong case for using insecticides to deal with malaria. Historically, it's been one of the most effective ways of eradicating malaria. So DDT was enormously successful in the Mediterranean basin and the southern US and so forth for eradication of malaria. So historically, insecticides have been really important. And at the moment, there's a huge program, global program of rolling out insecticide-treated bed nets across the world. And these are being shown to be highly effective. So insecticides are an extremely effective and actually very cheap way of getting substantial public health gains for malaria. So the question is, will we have problems with insecticide-resistant mosquitoes? And that is what happened in the 1950s when DDT was used in widespread areas at high level. The mosquitoes became resistant to DDT, and again in India and Sri Lanka and things. So there you have a very cheap, highly effective control tool, which all of a sudden is becoming threatened by the evolution of resistant mosquitoes. So the question we ask and others is that inevitable? Do we just need to sit by and watch while that happens? Is the best thing we can do to design new insecticides, find new chemicals that the mosquitoes are vulnerable to? Or can we use our existing insecticides in a way which might slow or even stop the evolution of resistant mosquitoes? And that's where we do think there are possibilities.
I mean, you start thinking about it, and wouldn't it just be great to have something that keeps them from reproducing? You keep them from reproducing, and then mosquitoes die out and they're gone.
Yeah, that's right. If you can keep them from reproducing and then they die out, that would be fantastic. But of course, any mosquito that's resistant to your insecticide that can keep reproducing will have an enormous selective advantage. Its offspring and grand offspring will fill the world because you're getting rid of all the sensitive ones, and the resistant ones are able to spread like wildfire. Right. So the trick to figure out is, how do we get rid of malaria-transmitting mosquitoes without creating the situations where this selection for resistance can happen?
Right.
And this is the way I think about this. I need to explain a couple of bits of natural history here first.
Please.
The first thing is that mosquitoes have a very tough life. They die very fast because their world is full of birds and spiders and the risks of blood feeding and so forth.
Are you trying to make me feel sorry for mosquitoes?
No.
just wondering.
But it is a neat thing about mosquitoes that they have a tough life. And they die very fast, so that most mosquitoes might live for 10 days at most, 2 weeks, something like that. That's about the average lifespan. The second fact is that malaria parasites take about 2 weeks to develop in a mosquito. So from the time that a mosquito picks up the infected blood from a person to the time that mosquito becomes dangerous, can transmit it back, that's 2 weeks. Okay, so if you put those two facts that I just said together, it takes 2 weeks for a mosquito to become infectious, but most mosquitoes don't live that long, then you get one of the great paradoxes of malaria, which is that most mosquitoes do not live long enough to transmit malaria. And the second thing that flows from that, which is the bit that, yeah, exactly. And the only reason there's a ton of malaria in the world is that there's a ton of mosquitoes.
Right.
Okay, there's gazillions of mosquitoes, so there's enough that survive long enough to transmit it. But most mosquitoes don't. So one of the interesting consequences of that is that if you are interested in malaria control, okay, not mosquito control, this is key, but malaria control, then you don't need to kill all the mosquitoes. You only need to kill the ones that have lived long enough to transmit malaria. And remember, most mosquitoes don't live that long. So you need to kill a small fraction of the population. That's the old mosquitoes and the ones with malaria.
Yeah.
And if you can achieve that, then you can get malaria control, okay? But the cool thing about killing these old infectious ones is that means most mosquitoes, most of the time, will have done most of their reproduction. So a sensitive mosquito that goes on to die will still have done most of its reproduction. Or another way of putting that is a resistant mosquito that survives this late life acting insecticide, it doesn't get much more of a reproductive advantage than does the sensitive one. Okay? Only a few of its babies are going to be produced that late in its life. Most of the 100 or so offspring it'll produce will be in the early part of it. Yeah. And so what that means then is that the selective advantage of being resistant to an insecticide, which is only killing a few old mosquitoes, is pretty weak. Actually, this is the same thing this is the same reason that we age. Okay, if you've got a gene in you that allows you to live from 80 to 85, let's say, it's under pretty weak selection because most of us aren't reproducing from between ages of 80 and 85. There are some people, but not most.
Right.
And
IVF is getting better.
IVF is going to change that. But even so, certainly most women aren't reproducing at that age, and rather few men as well.
Right.
So a gene that allows you to survive that long is actually under very weak positive selection. It's not got much advantage. So if there's any disadvantage to that gene, it just simply won't spread. And this is why we all age and fall apart. There is not much evolutionary pressure to keep us alive for a little bit long when we're old. Okay,
Right.
So the evolutionary pressure is weak. So from the point of view of making an evolution-proof insecticide, if you can make one that kills old infectious mosquitoes, then the selection against that will be much, much weaker. So all else being equal, it'll last a lot longer in the evolutionary arms race. It'll slow the red queen down enormously.
Right.
Okay, but here's the really cool part, and this is the bit I still think is very neat.
Okay.
If it's the case that the resistance genes come with a cost, so that it, for example, slows down the reproduction of the mosquito, does anything to the mosquito, if being resistant to an insecticide is a little costly process, and that's probably what it is because they've got to detoxify the compounds and so forth, spit them out. It takes energy, it takes biochemistry and so forth. If there's a little tiny cost to resistance, then just think about it. Any resistant mosquitoes, every single one of them is paying that cost.
Right.
And the only ones that are getting the benefit are the few that get to be old and old enough to transmit malaria, right? So the
Right.
cost benefits work out that everybody is paying the cost and only a few are benefiting. So tiny little costs are enough to offset the benefits of resistance, and they can stop the evolution altogether.
Wow.
Okay, so if you can get a compound or a process that can kill old infectious mosquitoes, and if that is associated with resistance costs in the earlier part of life, you solve the problem. And the thing about that, in principle at least, is that one single compound or product would work forever. So you would have stopped the red queen dead. You wouldn't need a pipeline of ongoing insecticide discovery.
Right.
You could stop it completely dead. So the question is not actually a theoretical issue of whether you can stop the evolution. It's an empirical question. Can you come up with a compound or a product which will kill old and ideally old infectious mosquitoes?
So you're killing the grandpa mosquitoes or grandma mosquitoes.
Yeah. Grandma mosquitoes. We don't care about the grandpa, just
right,
the grandma.
just the females. The females
That's
are the
right.
ones that go for the blood meals, right? And actually
That's females are the
transmit.
only ones that. That's right. They're the only ones that eat blood.
Yeah. So old grandmas in the mosquito world need to die. How do we
That's
do?
right.
How do we do this?
Ideally, the old grandmas who have the malaria parasites.
Yes. Ideally, only those. But how do you pinpoint them? How do you do this?
Okay. So there are two ways of doing this, which we are working on at the moment, and this is definitely experimental work in progress. I'll tell you the way which we definitely know works first. For some years now, we've been working on developing biopesticides against mosquitoes. So these are fungal entomopathogens, fungal pathogens that kill insects, and these are well known in the insect world. And they've been used for many years for locust control in Africa. And some years ago, my collaborator, Matt Thomas, came to me and said, well, maybe we could get this to work for mosquito control. So we have these entomopathogenic fungi. These are fungi that we can grow on substrates like rice. And you take the spores and you mix them with oil and you spray them on the wall. And it's low formulations, low doses, so you can't tell it's on the wall. And the spores just sit there and they do absolutely nothing until they contact an insect. When a mosquito crawls across the wall surface, hits one of these spores, then the spores germinate, they grow through the insect, and they kill it from the inside out. They eat it from the inside out. Now, so that's great. You can kill the insects, and we've shown in the laboratory that stops malaria transmission and has all the right properties. And a group in Holland has shown in central Tanzania that you can deliver these fungi in African huts to the mosquitoes. So in principle, it all works fine. Now, the very cool thing about these fungi is that they take some time to kill the female mosquito. And you can, by choice of fungal isolate, you can kill them sooner or later. So our proposition is that one of the isolates we're currently working on will have the property of killing the old mosquitoes. We've also shown in earlier experiments that the malaria-infected mosquitoes are more vulnerable to these fungi. So we can, in the laboratory, kill old infectious mosquitoes selectively. So we have with these fungus the prospect that one of them could work forever, that it would, in fact, never impose selection for resistance.
That would
So
be
that's
amazing.
the way we. Yeah, that's the way we already know that works. And the practical questions associated with that technology are not tied up with the fungal biology. They're tied up with issues of production, sustainability, environmental sensitivity, persistence on house walls, and so forth. And so we're pursuing that very aggressively.
Yeah. What are the, I guess, are there any downsides or human health concerns or even ecological concerns to this kind of a fungal insecticide?
Well, let me deal with human health issues first. Obviously, with any new public health measure, and this is true of vaccines, drugs, insecticides, anything, you need to check safety. Okay, human safety is vitally important, and you need to test that.
And we know
the reason
people.
We think this.
I was going to say, and we know people do have, or at least some people have sensitivity to certain fungal spores, and there are maybe concerns there, right?
So yeah, that's right. There are two possible safety issues. One is what you're suggesting there, the allergies against the spores and asthma type process, and clearly that's important. The measurements that we've done, or at least I should say my collaborator Matt Thomas has done in houses and buildings, it suggests that the amount of additional fungal spores that we add to a house is trivial compared to what is there, and that's especially true in African or tropical situations.
Right. Okay.
The other issue is whether a fungus could grow on a person, and of course, there's concerns with a lot of these areas where malaria is. There's a lot of issues about HIV and immunosuppression and so forth. So you could ask the natural question: could these fungi pose a risk to especially immunosuppressed people?
Right, and if they're a risk to the maybe immunosuppressed mosquitoes who are infected by malaria, then
you've clearly got to worry.
Yeah.
And we've got to test this obviously directly. The reason we're not too concerned, though it absolutely needs testing, the reason we're not too concerned is that these fungi that we are talking about, they grow at 28, 30 degrees at most. They are not happy at all at higher temperatures. So the sort of temperatures in human bodies, on our skin, in our lungs, in our noses and so forth, it's too hot for these fungi to grow. So we don't expect substantial risk, and there aren't too many examples of humans who've had infections at all with these diseases, like one or two ever recorded, and very, very difficult to find any significant clinical symptoms associated with those. The infections are small, transient, and so forth. So I think that safety side of things, I mean, it is not what keeps me awake at night, worrying.
Right, and then there's the other side of it as well, which is what is the acceptable risk. So with DDT is still being used in some parts of the world because the risk is acceptable, because malaria is so damaging to the human populations in those areas, that it's okay. They've decided DDT is going to work here.
Actually, so that's true, but it's an interesting question as to how much risk are we prepared to accept given the dangers of malaria. And I think in the modern safety, very nervous, risk-averse world, actually the risk we're prepared to accept is pretty close to zero. In the case of DDT, there's very little evidence that DDT is harmful when used in a public health context. It's clearly not great if you spray it across fields and saturate farmers and people. But if you spray small amounts on house walls, there's no evidence that it's causing human health risk there. Yeah. So the DDT thing is not actually a cost-benefit calculation of a little bit of risk versus a lot of benefit. It's actually a perception that it's zero risk versus a lot of benefit. And I think our fungal approach has to hit something pretty close to zero risk. And in insecticides that run a risk of killing anybody simply aren't actually going to be used.
Yeah.
But I am pretty confident. I mean, the fungus is approved for use in locust control in Africa and in Australia, where it's sprayed on very large areas. And under those situations, that people have done the testing and the ecotoxicology testing and all of that side of things. So it needs to be tested, but I'm definitely not losing sleep over it. The other thing you did mention, though, which is something that is typically, if I might say, raised by Westerners, is the issue of environmental safety and what damage might be done if you set out to attack mosquitoes. The standard question is, if we got rid of mosquitoes, what's going to come in instead?
Right, and the question always is, people always wonder, what good are mosquitoes? Are they just food for spiders and to bats and insects? Is that all they do in the ecological scheme of things? Do we really know?
And I think we probably don't really know.
Yeah.
And so you could say, well, what would be the effects of removing mosquitoes? And there have been a few bad cases of that where species of mosquitoes have been removed by insecticides, and then other species have come in and transmitted malaria, perhaps even worse.
Right.
Not too many examples of that, but there are a few. One of the nice features of our approach is, remember, we're only killing the old infectious mosquitoes. And
The
there's
old
hardly
and
any
the
of
sick. Yeah.
Exactly. The old and the sick, and there's only a few of them, right? Like a few percent. So we're going to leave 95% of the mosquitoes behind. So we're not creating a big gap. We're not creating a vacuum into which bad things can pour.
Right.
But if you ask an African housewife what she makes of mosquitoes, and does she worry about, what would be the outcome of losing them? Actually, they're living in her house. They're in her ceiling. They're disgusting. And, they and everything else in the ceiling, she would be quite happy to get rid of. It's blowflies and spiders in your ceiling. It's just not a,
It's not a happy environment.
Well, it's
Not
an ecological it's an ecological concern of Westerners, and her children are dying of malaria.
The concerns are completely different in that case. Yeah.
Absolutely.
Yeah. Are you looking at you're looking at this fungal intervention? Are there any other ideas that are out there that are kind of looking at the same life stage of the mosquito and controlling that, or looking at other possible directions to be able to control the malaria parasite in this kind of evolution-proof way? Are there any other ideas that you're kind of competing with?
Well, we are doing some work with chemicals, asking whether we can find compounds, either existing or new compounds, which would kill, as you say, the old and the sick mosquito. And the argument here is that the old and the sick mosquito is going to kill. So maybe our existing insecticides, if we just diluted them down, just used them at lower concentrations, maybe that would have precisely the same effect. You would, if you diluted it to the point where the young, vigorous, active mosquitoes weren't dying, but the old, senile, sick mosquitoes were dying. If you got it to a point where that's only toxic enough for the weak, then maybe we could selectively remove the old ones without affecting the young ones, and then that would be evolution-proof. So we're working on that at the moment. We're also working on whether there might be other compounds which are currently thought of as being too insufficiently toxic to be insecticides, which actually might work just fine if you sprayed them only intending to kill the old ones. There are one or two other ideas for how to kill old mosquitoes, and these involve viruses or bacterial infections of the mosquitoes, and there are people actually actively working on those. And either their approach or our approach could potentially lead to an evolution-proof solution to the insecticide resistance problem.
Yeah. Does that come down to like which ones are going to be more success? Which of these solutions might be more successful? Do you think it's going to come down to cost effectiveness? Is it is a fungus?
Yeah.
Yeah.
Yeah. I wish. I actually think it's going to come down to who is able to persuade the funders to support enough work at the right time to take things further forward. So some of the work on the viruses or the bacteria is involving some very expensive molecular biology, but potentially is very high payoff. So it requires more upfront science, but it's more high payoff. Our approach is probably easier to implement because it's not that different from what's happening already. We'd be putting insecticides on bed nets or on walls. So it's the same sort of approach. It's just that we would be using different compounds or the fungi or so forth.
Right.
But I increasingly think in this business of trying to deliver solutions to world problems, especially world problems associated with, cheap healthcare products in the third world, it's luck of the draw in the funding issues actually, which is going to be a major determinant of who actually gets there. On the other hand, if there's suddenly a big problem with insecticide-resistant mosquitoes, as people are talking about now in sub-Saharan West Africa, if the bed nets start to fail, then people are going to be looking for new solutions in a hurry, and that's going to change things. And that is my expectation, that in the short to medium term, resistant mosquitoes are going to overwhelm some of these currently very effective programs in bed nets, and then people will be looking for new solutions. And obviously, I very much hope that our ideas can contribute to that next generation of solution.
Yeah, well, it seems like the logic behind the direction that you're thinking, it seems like good logic. It seems like it would work. So, hopefully it will.
Yeah.
Hopefully it can go into practice and actually be tested.
Yeah, I think the logic is fine. and you can show in the computer that it can work. The question is a practical one of, can you find the right compounds? Do the fungi survive long enough? Can you transport them? Do you need a cold chain? Those sorts of issues. I think the logic. I haven't heard any sensible criticisms of the logic. The
actual application.
The issues are practical. Yeah, absolutely.
And so are you interested in working on those practical application side of things, or are you going to stay within the realm of working on the evolutionary science, working on the, how are these organisms evolving? What's happening? Where can we press certain buttons to make a lever move this direction? Where can we affect things?
So that's a really interesting question. If there's somebody out there now listening who wants to take this stuff and make it happen, I'd be absolutely delighted if they did that.
Yeah.
Because actually the implementation stuff is really, really hard work and politically difficult and all of that. But it's not conceptually so exciting. So it's not really fundamentally interesting evolutionary biology. So if in my best of all worlds, we would do the groundwork to prove principle, and then somebody else would run it and implement it. At the moment, though, the ideas are actually so far outside the existing box that other people are not picking this up and running with it. So we are doing the implementation work. And I would like to get the level of implementation work, hut trials and then maybe village trials, stay involved in that to the point where the data are convincing enough that other people start to run with it, people more skilled than I in the field trial side of things. But at the moment, I don't see anybody else picking it up, so my collaborators and I running with it. Matt Thomas, who's also here at Penn State, is especially getting involved in work in India, and we hope to take that further forward. Just met today with people who are interested in doing work in Mozambique and things. So, we really are trying to do it. But I have to say, from my personal point of view, it'd be a lot more fun to stick with the computer models and the lab work.
To each their own, right?
Absolutely.
Everybody's got their own idea of what really inspires them and what keeps them going, and the moving out of the lab and into practical application really is a completely different world. It's different motivations, I think, to keep you going in that.
Yeah, that's right. I suppose what's interesting from my point of view, the way I see the sort of very big vision here, is that if you think hard about how natural selection is working and understand modern evolutionary theory, the question is, does that lead you to new ideas, new ways of tackling evolutionary problems, which potentially in this case can save millions of lives? And it would be very nice that at the 21st century, new and proper ways of thinking about evolution can lead to novel solutions. So that big vision, I would be, I am very keen to stay and keep pushing that forward, as opposed to the sort of old 20th century view that we just keep discovering new drugs or new insecticides. We just keep looking for new compounds,
right?
And then watching while they fail,
right? Because it's just going to keep going like that over and over unless we change the way we think about it. It's not going to end, and everything will escalate.
Yeah, actually, it's an interesting question as to whether it'll escalate, but you certainly need to have a certain speed of research and development to find new drugs and new insecticides. And that process of that pipeline, that treadmill, has to be kept up and funded even when there's no problem right now. So, as you said before, artemisiums, magic wonder drugs against malaria, they are very effective, a fantastic drug. There are now the first signs of at least drug tolerance amongst some parasites. So then you think, well, okay, there's artemisiums working right now, but it may not work. They may not work in 5 or 10 years' time. Wow, your studio's falling apart.
I know. Something just fell down behind my head. I will get that in a minute.
Okay, let's hope it's not an earthquake out there, is it?
I know what's going on. I am in California.
Yeah, you are. Well, it's nice and still in Pennsylvania.
Yeah.
So you need to keep this pipeline of drug discovery and insecticide discovery going, and there has been figures recently of the sort of costs involved in that, and it might be in the order of $1 or $2 billion a decade to keep that process going for anti-malarial discovery. That's just the research and development costs, not the implementation, just the research and development costs.
Yep.
So, can we keep that up indefinitely until malaria is eradicated? I don't know. And each new drug or each new insecticide is probably going to be more expensive because we've already discovered the cheap ones. So, and people die in the time when you're swapping drugs or you're swapping insecticides. So, and not to say that we should stop the discovery, but I am interested in the question of whether we can shape evolution to be a bit more productive from our point of view. And, of course, at some level, we're not the only ones saying that. I mean, we all would like to reduce the use of antibiotics in the community, right? We don't want antibiotics to be freely available. We don't want viral infections treated with antibiotics. And so, as a society, we try to reduce antibiotic use. That is an enormous evolutionary experiment. We are trying to slow down the evolution of resistance against antibiotics by reducing use. So we are already trying to manipulate evolution.
Right.
And that's, for me, that's a no-brainer. My question is whether there are other less obvious ways of doing it that, for example, to do with how you use the insecticides like I just talked about, or how you use drugs in a patient.
Yeah, and that's something that slowing down. That's something that just because you're not using the drugs as much, the organisms are not coming into contact with them as much, and so the opportunity for resistance formation is decreased. Is that right?
That's precisely it. So the reason that the resistant mosquitoes or the resistant parasites, resistant viruses or bacteria have an advantage is only when there's drugs around. Okay, if there's no drug or insecticide pressure, you don't get resistance.
Right.
Okay, that's clear. It is clear. So, but that immediately leads to ideas that are quite non-intuitive. I've already mentioned the insecticide case, but it's interesting that at a population level, we say for antibiotic use, let's not use any more than we absolutely need to. That's good advice. But when it comes to the actual going to the doctor, the physician, when you get your antibiotics from the physician says, take all the tablets in this drug course, even after you feel better.
Yeah, keep taking them.
That's right. And so long after there's no clinical need for you to keep taking the drugs because you feel better, you keep hammering away at those pathogens. You keep taking the drugs and you keep killing all the sensitive ones. And, hit them hard, hit them long, get rid of every single sensitive one. Well, who's going to take over? The resistant guys. You give every resistant parasite an enormous advantage when the susceptible ones are being removed by drugs. So what we're working on at the moment in the case of the antimalarials is the question of whether or not it's a good idea to keep taking the drugs for the full drug course so that, in fact, you've got rid of every single susceptible parasite and then given every possible advantage to the resistant ones. So we are right now testing what I would take as one of the most sort of fundamental one fundamental orthodoxies in how you should take drugs. I should say quickly before any of your listeners out there are suddenly going to stop taking their drugs, that is not what I'm suggesting at the moment. We are still working at this. And I see on Dr. Mom on the chat show has already come up by saying you get more drug resistance by not taking the full dosage.
And that's the popular view, right?
That's right. And that is precisely the question I'm asking. Is that really true? I agree everybody says it's true, but what tells us that that is true?
Is there evidence? I mean, what is the evidence that has led this to be the popular story?
Actually, I'm not sure there's any good evidence that that's the popular story. It goes back an awful long way in biomedicine. It goes back to the early days when Fleming discovered penicillin, and he wrote in the New York Times, actually, during the Second World War, he wrote an article saying penicillin is a magic drug, but it is not going to work forever because if I lower the doses in my Petri dishes, I can get resistant bacteria. And so it seems to have started there, the idea that you need the full long-course chemotherapy to kill every single thing that's there so that there's nothing left to mutate to resistance. You with me? So if you've got if you're just filled with susceptible parasites, and if you can kill them all, then clearly there's going to be no resistance, right?
Right, you've got them all.
But you've got to make sure, you got to make sure you got them all, and you got to make sure there wasn't even just one single parasite there that was resistant. If there's one single parasite that's in your body and you start hitting it with drugs, that guy's going to survive and everybody else is going to get removed. So who's going to have the advantage? The resistant one. So there's this drug treatment has this two-sided effect. Okay, it reduces the number of parasites that could mutate to resistance. All right, so you have fewer parasites. There's less chance of resistance arising in the first place. But if it does arise, or if you got resistant parasites from somebody else, then treatment of a very strong bang, 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 that is going to get rid of every single sensitive competitor and give the resistant parasite the biggest advantage it could have.
Yeah.
You with me? So drug treatment has this two-sided effect. And we don't know how those two things balance out in any case, in any one case. And certainly in the experiments that we're doing at the moment with malaria in mice, actually it definitely comes out that the best thing to do is to treat less than the current recommendations.
How interesting. That's really interesting. So I'm looking forward to hearing when all of the research is done and you've actually, you've got a complete story to tell, because it's not the doctrine at all. No,
and I also absolutely want to emphasize this is research in progress. We're working these ideas through, but I think the orthodoxy, the standard thinking, is not well supported in sensible evolutionary biology. There may be special circumstances when it applies, but it is not going to be generally true. And the question is to work out when it applies or when it doesn't. Right. For instance, chloroquine used to be the magic wonder drug against malaria. Chloroquine is now useless in many parts of the world because of resistance. Nobody in Africa got chloroquine resistance from something that happened inside their bodies. They got chloroquine resistance from mosquitoes. Mosquitoes transmitted resistant parasites from somebody else. Yeah. And so the best way then, if it's got into your body, of getting the chloroquine resistant parasites to transmit from you onto the next person is to treat with drugs, get rid of all the sensitive parasites and just give the resistant ones the advantage. So we need to work this out. It's an open question. It's absolutely a research question at the moment. But I think the sort of experiments that one needs to do on this, starting off in petri dishes and computers and mice and maybe in due course primates, needs to be stepped up gently and then tried very delicately in small number of humans to start with and develop. We need to work this out, though.
Yeah, it's something that instead of just telling people something that's a blanket answer for everything, there should actually be some evidence behind it. So I'm glad you're working on it. That's precisely my feeling. And I think the more I think about this is if you just think about the number of different roles that insects and birds and things have in forests, no one generality would fit them all. Why do we have one generality to fix all bugs? It just doesn't follow. Right, and not every bug has the same life cycle or life history. Not every bug has the same, lives the same length of time. Not all of them reproduce at the exact same stage of their life. Yeah, so all of those factors.
And exactly, not all the drugs work the same way. Not all the drugs target the same things. The transmission is different. The immunity against different diseases varies. There's a whole range of things, and we need to work this out in any particular case before we actually know what's the best thing to do.
Yeah. And the complexity just seems like as we learn more, even as we start using scientific principles, evolutionary theory to be able to guide our thinking, it just seems like the complexity is increasing of the problem at hand. So where once we could say, okay, this drug goes, this regimen, this long, whatever, now it's specific for each particular instance where it might have to be given. How do you start looking? Do you start looking at the people who are also taking drugs that are involved in this whole story of parasite, insecticide, parasite, anti-parasite drug?
Yeah. Well, so that is one way to go forward. I think and there are people doing that. There's some very interesting work at the moment coming out of Malawi where people are examining the clinical outcomes for people who take drugs or don't take drugs in an area where there are many resistant parasites around. That is definitely one way to go forward, and I think that's very important work. It's also very difficult work, and it's usually ambiguous because you can't, in an ideal world, you want to randomly allocate who gets the drugs. Yeah. Okay. And you obviously can't do that. Sick people need the drugs. Some people won't take the drugs because of some objection. Yep. And they'll have other effects. They'll have other infections and so forth. So the people side of things is actually pretty tricky. But I think that's where we're going to have to go eventually. I guess I feel at the moment that what we're doing is sufficiently controversial that we need to prove concept in animal models first. The veterinary situation where people are treating pigs and horses and cows and sheep and poultry, that's a situation where we really could do a lot of work on a large scale in this to try and figure things out better.
Right, as opposed to where currently antibiotics are just being mixed in with the animal feed just to maintain, it's just a maintenance drug currently.
Yeah.
That's something that could definitely be investigated and probably fixed.
Yeah, there's a really interesting question. There is a really interesting question about why the antibiotics in animal feeds still work. Why is it that putting them in the feeds is still giving yield gains? The farmers still like doing it. Why is that? If there's antibiotic resistance around, surely that antibiotic should have failed by now.
Yeah.
And I think that question of why they haven't failed in an agricultural situation, they've almost been evolution-proof in an agricultural situation. They've worked for many decades. Why is that? And we don't know. And if we could get that to work in hospitals as well as it works in farms, that'd be a cool thing, wouldn't it?
That would be a cool thing. Most definitely. Wow, these are such interesting questions. I'm so excited about the implications for all of this research. Now, on the human side of things, there was a paper in PLOS that you published last year where you asked the question of, is damage control more important than parasite control? What did you mean by that?
Well, this is tied up with the question of when hosts are naturally resistant to parasites. What do we actually mean by that? And we all know there are some individuals who are more inclined to get flus each year, or who are the ones who always get the playground diseases. And one of the questions in biomedicine, of course, is what's causing that variation. And some of the variation is clearly the way people lead lives, but we also know that some of the variation is tied up with their genes. And we've then got the question of, well, when people have genes which make them better defended against infectious disease, how are those genes working? And typically, not always, but typically immunologists are the ones working on these problems. And typically immunologists are interested in the mechanisms by which the incoming pathogens, the bugs themselves, are being killed. This is kind of like, in a military sense, the mechanisms by which one knocks out missiles or tanks or the oncoming troops. So actually taking out the oncoming enemy. But there's also the possibility that there are mechanisms involved with being able to better withstand the damage these things cause. So instead of just attacking the bugs, you could put up with the damage they do. You could get better at repairing it or whatever. So in that case, it would be kind of like, an aircraft carrier is a very hard thing to sink, because even if it's being hit by a lot of bullets and gunfire and so forth, it's a big, solid thing, and it's very, very hard to take it out. So maybe there are mechanisms of host defense which are tied up with that sort of being more robust, being tougher. And the interesting thing about those sort of mechanisms, and I think here of tissue repair, mechanisms for replenishing red cells and things. The interesting thing about those mechanisms is that they're improving the health of the body without attacking the parasite, and that stops the red queen. That means that those mechanisms are not having a bad impact on the parasite, so that parasite doesn't need to evolve back against them.
Right.
Okay, so you're not attacking the parasite. The parasite is allowed to get on with what it's done, but you're stopping it doing damage. Now, to the extent that that is separated from the fitness, the evolutionary success of the parasite, then that means that the parasite doesn't have to evolve back against that. So you could get evolution-proof defense mechanisms. We're actually not the first to say this. People have said this in plant biology, that maybe instead of trying to breed wheat that's resistant to rust, that can kill rust or fungus infections, maybe we should be breeding wheat that's more robust to fungal infections. So it just can put up with the fungal infections.
Yeah, so in the first place,
and then
I was going to say, so in the first place, it either doesn't get infected, or if it does get infected, it doesn't have the same kind of processes that go on. It's stronger. You can fight it better.
It puts more effort into just being tough and vigorous, and therefore those mechanisms are going to continue to persist because there's no evolution of the pathogen to go around them. Now, this is a controversial idea as well, but it's a very interesting one, that maybe most of the things that defend us against disease are of that sort, just make us more robust to their infections. And we know we get lots of infections without getting sick. So maybe most of the defense mechanisms in our body are of that sort, rather than of the antipathogen sort. And that's an interesting idea. Maybe we could try to work on that as a new way of dealing with infectious disease in humans and in agricultural animals and plants.
Yeah, it sounds really interesting. The thing, usually our immune systems work very well, but then there are the times when our immune systems go a little haywire, and we have autoimmune disease, or we have situations where, say, the inflammation from an infection can actually cause damage in your body and cause other downstream effects that are deleterious to your health. That's not necessarily going to be good for your survival. That's
right. And I think that is one of the interesting evolutionary research problems of the 21st century. Under what situations is it worth the body doing self-harm? I mean, that's essentially what inflammation is, and a lot of the autoimmune diseases you're talking about, the reason we die of flu, if we die of some of these, the swine flu type things, those typically are tied up with malfunctions of the immune system. It's not like we're eaten alive by the virus. It's the body's reaction to the virus that's killing us. And it's a very interesting question as to whether we have those apparently deleterious overreactions, because in most circumstances they are very beneficial. And that's my bias. If you get malaria parasites, you can't muck around. You've got to kill them.
Yeah.
So maybe the body runs a rule like that all the time and occasionally gets it wrong, and maybe that's rare enough. So I think that's a very interesting question as to why we get that sort of immunopathology. It may also be quite rare in the scheme of things. It might be a function of Western lifestyles, and it might be that immunopathology, self-damage is a lot rarer where you have a lot of worms, where you have a lot of infections going on that are modifying the responses in ways that are perhaps what would have been more ancestrally natural.
Right, and that's a popular idea right now, isn't it? The idea that
that's right, that
in Western society where we're very clean and we don't have a lot of internal parasites and other nasties, that our bodies are more likely to have these inappropriate immune responses.
That's right. So worms, for example, parasitic worms, the big ones that live in people's guts and sometimes bloodstreams, those, if you think about those are very big organisms. I mean, they are the size of earthworms at times, and they're living in a gut or in a bloodstream, and they're not being rejected by the body. Whereas if we try and do even another human organ into our body, it gets rejected. So these worms are living without being rejected inside our bodies for often decades at a time. And the way they do that is by pumping out substances which immunosuppress locally. Right. Like we do for an organ transplant. And those substances might mean that normally our immune system is damped down by these worms. Everybody would have had worms once. And so maybe the response is damped down. So our immune system fights back by getting more sensitive. And the two pressures come together and you get something sensible, balanced in the middle.
Right.
But then you take the worms away and hey presto, you've got
really hyperactive immune system.
That's right. That's right. Yeah. Yeah, I think that's a very, very interesting and entirely plausible hypothesis, which does lead to interesting new ways in which we might try to manage some of our inappropriate disease responses.
Yeah, all of this is so interesting.
Well, that's because, Kirsten, everybody should be an evolutionary biologist. It's really
everybody,
and everybody should be one working on infectious disease. I don't know why anybody does anything else.
Because there are other interesting things to do in the world. There are.
Like what?
I don't know. You could study birds again.
Yeah, birds are very interesting, and that's true. But I think, human misery is also one of those things we should try to work on alleviating. Look, lots of evolutionary biologists study birds. Hardly any study infectious diseases. It's changing, but it's in proportion to the number of issues. There's a lot more evolutionary biologists studying birds and trees and insects than there are evolutionary biologists studying infectious diseases.
And infectious diseases are probably one of the biggest issues we're going to be dealing with as we move forward in our global future of more and more air transport, more and more, different kinds of people mixing all over the globe. Things are no longer going to be stuck in their little pockets. So we have the flu coming from Southeast Asia. More and more likely that a really bad flu will get out of there. Malaria coming out of Asia and Africa. And we have the Anopheles mosquito down in San Diego, the southern part of California, and even the southern United States. The malaria carrying mosquito is here. So these are things that are going to be more and more pertinent.
Yeah. That's absolutely true. I wouldn't be too worried about getting malaria in San Diego. The mosquitoes are there for sure, but the mosquitoes are in Pennsylvania too. And the mosquitoes are in England, and there used to be lots of malaria in these areas. Yeah, they're all over. But what does it now is, of course, we've got nice houses with good walls and mesh screens and good drugs. And so it's not often that a mosquito in San Diego, even if it was to bite a person with malaria, would actually go on to bite another person and pass it on. You know, you get the odd case, but not too many. But you're right about the general issue that diseases will move around the planet much faster now. And the other side of it is that we are disturbing the environment much more, so that the people are coming in contact at a much greater rate with things that we previously wouldn't have done. So there's a lot of potential for disease transfer from animal reservoirs, and there's a lot of potential, once it's got going, for very rapid transmission around the world. And HIV is a classic of that. It probably was a persistent thing coming into human populations but not getting very far, and nowadays, of course, it can go global in no time.
Yeah. This has been really, really a fascinating conversation. I don't want to keep you too long, but one thing I did want to get back to, no flash on this machine. Will it open? One thing I wanted to get back to was the kakapo. because the kakapo is it's a flightless bird, and here, let me see if I can bring this up. So this is the kakapo, and the kakapo is a flightless bird from New Zealand. And this is a bird that you spent time studying.
Yeah, when I was an undergraduate, I was very fortunate to work for several seasons on these birds. And these birds are some of the most amazing birds in the world. So they're the largest parrot. They don't fly, they sort of drift, they glide a bit, but always with a downward motion. They breed only every few years. The males contribute nothing but sex to the females. And then the females raise their offspring in forest floors, in holes in the ground. And these eggs, when they hatch out, are pathetic little chicks that can't move, and they need to be fed for months on end. And it's a very different lifestyle from the average bird. Now, that would have worked fine in a prehistoric New Zealand, but nowadays there's cats and stoats and rats and so forth. So these birds are extremely endangered. And to give you a feel for how bad they are in terms of adaptation to the modern world, they actually smell nice, and they sort of smell like hay, fresh hay to us. And I can smell them when you get close, and often you can smell them before you can see them.
Wow.
So imagine what they're like for a cat or a rat or a stoat. Very easy to find.
And when they see predators.
Exactly. And then when they see when the birds see a predator, they just freeze. So they don't move at all. So they sit in front of a predator smelling sweet, saying eat me sort of thing. So really a very bad environment very bad lifestyle for a modern environment. The New Zealand government and lots of New Zealand corporate sponsors have spent a lot of money preserving this bird, working very hard on its conservation. And actually the future is pretty bright, a lot brighter than when I worked on them, when we really thought we were watching an extinction in process.
Wow.
And the reason Kirsten's mentioned this at all is that when I was working on these, I used to worry about how could it possibly be that evolution would have produced something so completely and utterly useless.
Yep.
And it does make perfect sense in a world where there were no predators, because they didn't have to worry. The only predators were avian ones, which don't smell that strong. The breeding system, where you have them just coming together for sex and the male not helping, that has actually evolved in quite a few different situations, and it can be one of those things natural selection favors under the right conditions.
Yep.
So I think we actually have, from modern evolutionary biology, an explanation for this very unique and quite fatal lifestyle in today's world. And it was that got me thinking, well, there must be something in this evolution stuff. If we can explain something as weird as the kakapo, it should be really easy to deal with things like antibiotic resistance and malaria and stuff.
That's right. You tackled the hard question first, and now it's getting into the easy stuff, right?
I wouldn't say that. And it is, to be fair, kakapo are so endangered that nobody can do any science on them other than science associated with the conservation. But at least in other situations, we understand those breeding systems, we understand those lifestyles. And so, that's one of those examples which I think if there's an intelligent designer, he really designed or she really designed those birds badly for the modern world. There was a complete lack of foresight. And we understand lack of foresight.
No planning.
No planning. That designer really did not do a good job with those birds. But natural selection is not looking at foresight. Natural selection is not looking ahead. And so that's what you get.
Yeah, you end up with an animal that is badly adapted to the situation in which it currently lives. And there are probably many more animals around the globe that are in situations like that currently, or will be in situations in which they are poorly adapted at some point in the future. And it could even be us.
Might even be humans, yes.
It could be us.
Yes, indeed. Indeed.
Thank you so much for your time today, Andrew. This has been a fabulous episode of Dr. Kiki's Science Hour. I've enjoyed learning so much about infectious diseases, about this way of thinking about disease in an evolutionary perspective from that kind of a framework. So it's been very helpful. Thank you.
Sure. Thanks a lot. Great fun.
Yeah.
Thanks very much.
And if anybody has would like to find more information about battling the Red Queen or the evolution-proof drugs, you can go to thereadgroup.net. And that is where you can find research that Dr. Andrew Reed is working on currently, and papers and things that he has also done through the past, so you can get more information there. I'm Dr. Kiki, and this has been the Science Hour. And if you would like to follow me elsewhere, you can find me on Twitter. I'm at Dr. Kiki, @DrKiki. You can also follow me online. My website is drkiki.tv. And you can get this show through iTunes if you go looking for Dr. Kiki's Science Hour in their Science and Medicine directory, or also in the Twit Network directory. And I'm trying to think if there's anything else that I've left out here. Oh, yeah, This Week in Science, my other radio show, twist.org, Tuesdays. That's the Tuesday one. So there's always more science coming out of my part of the world for you. And I will see you next week. We're going to be talking with Dr. Karen James of the Beagle Project. It's a project to get a replica of Charles Darwin's ship, the Beagle, on a voyage to the Galapagos to do some evolutionary science. So we'll have maybe some fun sailing around the oceans next week. Thanks for tuning in. Peace out.